would you be able to tell me what the standard view of evolution is today?
Hmm. Yeah, so most of us who work professionally on evolution, um, think of it as a process where there's two kinds of phenomena that are important. The first is um, that life produces variation in organisms, that is, a variation in their outward appearances, in their traits that makes them able to survive and reproduce in the environment. There's different kinds of ways in which such variation is being produced. For example, one is called DNA mutation, and that is called recombination. And the other process is actually a class of processes um, that filters, if you will, this variation and keeps only the useful stuff. Okay, and that's what's commonly known as natural selection.
And what is that? So with natural in the processing, the filtering,
Hmm.
is that animals the ones who survive, the ones who reproduce successfully?
Yeah,
Um,
yeah, so the filtering can actually take, many different forms. So I'll give you an example from microbes, which are the kinds of organisms that we work with. Most of the time, when we work with microbes in the laboratory, um, we um, we study how fast they grow and divide. Okay, they grow and divide, very fast by human standards. One generation time of a microbe is somewhere between 20 minutes and a few hours. Um, and let's imagine you have a big population of such microbes that have different genomes. So there's variations of the same genome in that population. Some may grow a little faster, others may grow a little slower, right? Um, and The ones that will grow faster will very, very rapidly outcompete the ones that grow more slowly and will eventually drive them to extinction. Okay, so that can happen, that would typically happen within a few weeks or so. So that's one way in which this filtering happens. So that's an example of selection. In this case, selection in the laboratory, not so much in nature, but in the laboratory environments we create. But even in the laboratory, this kind of selection can take place in different ways. So, for example, if you let microbes starve for a long time, and I mean days and weeks, so basically you just don't provide them with nutrients, um, they hardly divide anymore, okay? Because they simply don't have any food to grow on. Um, and so what happens in that case is that some of the microbes are um, better able to deal with that starvation and survive it for longer than others. Okay? Um, and the ones that don't survive for very long, they die, and there the molecules in them become food for the ones that survive, right? So that's also a form of selection, but it's a completely different form of selection where there's nothing more about cell division really going on. It's just about survival of starvation. So within the same kind of organisms, selection can act in very different ways depending on what kind of environment they're exposed to.
right. So there's like, yeah, interesting. And there's different examples. I mean, in one, it's like one species of a bacteria might be replicating fine, but it's just there's so much slower than another species, so then they're outcompeted. On the other hand, that last example is like they're not even really reproducing. It's just sort of
Mhm.
like in that environment, you just need to survive. Um, and so there's, and it's interesting. One thing I wanted to ask as well is on the DNA mutations, right? How does variation occur?
Mhm.
Tell me, I'm something I try and understand, and I
Mhm.
would like if you could explain it. So I mean, how, because where do the mutations have to occur? Like how often is DNA being
Mhm.
mutated,
Mhm.
and how do mutations actually occur?
Okay, so well, there is two kinds of causes of mutations of DNA mutations. One is external causes, so these would typically be, let's say, high energy radiation, like from UV light. When your skin gets exposed to UV light, DNA mutations occur because the DNA in your skin gets irradiated. But there's also chemicals in the environment that are responsible for mutations. So that's external causes. But then there's also internal causes. For example, our metabolism produces chemicals called free oxygen radicals and related chemicals that damage DNA. So that's an inevitable byproduct of being metabolically active. When a cell consumes food, it produces these molecules and they damage DNA. So cells have actually um, developed or evolved a slew of mechanisms to control this kind of damage, but it cannot be completely eliminated. Now, how often do mutations occur? That depends very, very much on the organism. Since we were just talking about bacteria, let's give you a number. Um, so the we speak of the genomic DNA mutation rate. This is basically the probability that a single DNA letter in the genome of a bacterium is changed in a single generation. And that rate is about 1 in 1,000 per generation. So what this means is that we'll have to wait 1,000 generations, 1,000 bacterial generations to see a single letter change in the genome of a bacterium like Escherichia coli, which may mention again because a lot of people, including people in my lab, work on it a lot. It's sort of a workhorse organism for laboratory evolution studies. So 1 in 1,000, that doesn't really sound like very much, but since, remember, bacteria divide extremely rapidly. You might, in a single petri dish, you may have billions of bacteria and millions of cell divisions and generations that took place. Um, and Therefore, these kinds of small probabilities add up, as you would actually typically in a single petri dish have a genetically diverse population of bacteria just as a result of DNA mutations that happened on that petri dish. To give you a different organism, humans. In humans, the rate of what we call point mutations, so these are mutations that change a single DNA letter at a time, is about 40 such mutations every generation. Okay? That sounds like a whole lot more than Escherichia coli, but actually you also have to take into consideration that we have much, much larger genomes. Right, so E. coli has a genome of about 4.5 million DNA letters. We have a genome of about 3 billion DNA letters. So if you consider that, then 40 changes out of 3 billion letters every generation is not actually all that much.
When mutations occur, do they have to, if they just occur in like a skin cell, or any, is it what cells? So because I assume it would have to be in the germ cells, like the reproducing sort of cell. Is that right? So where do mutations occur exactly?
Well, mutations occur everywhere all
They
the
occur.
time, right? So they occur especially in tissues and cells that are dividing actively. What I didn't tell you is that a lot of the DNA damage that comes from internal causes actually has to do with cell division and DNA replication, because whenever DNA gets copied, then the the biochemical machinery that is responsible for that copying and replicating makes mistakes. Okay, um, so you see preferentially mutations in dividing cells, but also in other cells. Some of the very actively dividing tissues is, for example, our skin, but also the inner lining of our intestine. Right. This is part of the reason why cancers in these tissues are particularly important. Now, then you made an important distinction, or you hinted at a distinction. There's the distinction between germ cells, so the cells that are inherited, um, and cells that are not germ cells. We call them somatic cells. Mutations happen in all of those cells. In somatic cells, typically the consequences are bad. Think cancer. In germ cells, that's the kind of genetic changes that get inherited from one generation to the next. Um, and germ cells, especially sperm, are extremely actively dividing, right? And so therefore you also have a lot of mutations occurring in there. And when I talked about this rate of 40 new mutations per generation in humans, that refers actually to the germline mutation rate. So this is the kinds of changes that do get inherited.
Do the sperm cells have more mutations than eggs then? Is there more variation in the male germ cells?
So now you can't quote me on that because I'd have to look this up, but I'm pretty sure that it is the case because of their actively dividing, right?
Mm, interesting.
But there's also um, oocytes or egg cells. They are basically made, if you will, very early in the life of a person. in the life of a woman, and then they're resting for a long time, and this resting also has potential to cause damage, right? Um, it's just that the kind of damage that you typically get in egg cells is a bit different genetically from the kind of damage that you get in sperm cells. So, for example, I've spoken only about point mutations, that is, mutations that change single letters. But there's other kinds of mutations that are also important that I didn't mention at all here, and those are what we often call structural changes in genomes, where, for example, an entire chunk of a of a chromosome might get lost, or gets duplicated, or gets inverted in its orientation in the string of DNA. And something that is fairly well known is that certain kind of chromosomal changes tend to get more frequent with female age. So that means the oocytes are more susceptible to those kinds of damages. But it doesn't mean that you don't get damages accumulating in sperm over the age of a male, over the lifespan of a male. So there's in fact a lot of damage that's accumulating there as well. And this is part of the reason why your children's, if you're older and you have children, either if you're a male or a female, your risk that your child, that the risk that your children have genetic diseases increases with age because of
Mm.
accumulating damage.
I kind of wanted to ask about, because you talk here about DNA, and it's interesting that DNA is, our understanding of it is something that's more recent in scientific times, that we understand that it's the mechanism of evolution. Or so what do we what do we think about evolution before DNA, and we understood about that, and how has it like changed afterwards, and is
Mm-hmm.
DNA the only way that evolution works?
Mm-hmm. Well, it's let's perhaps, to answer the question, go all the way back to Darwin's time. Darwin already knew about variation, because variation is something you can observe all around you. If you look inside a forest, not all the trees are identical, and same holds for, I don't know, a meadow of flowers, and various animal species, and even within species you have lots of variation. So Darwin was aware of that. Um, but he considered the question where variation comes from a mystery. He had some ideas about it, and most of these ideas turned out to be wrong. Um, but basically, he just did not have all the information that people just 80 years after him or so had accumulated about variation. And so that's something that the cause or the origin of most of the variation that is out there um, was completely unknown to Darwin, and it would take, well, where to put that date? It was demonstrated in, I think, 1942 or 1943 that DNA is actually the material basis of inheritance, and then it would take another 10 years or so, until Watson and Crick discovered the famous double helix, that is the chemical structure of DNA. And it was only then really that people started to get the handle on what DNA mutations are really about. So what we have now that Darwin didn't have is a mechanistic understanding of the origin of variation. You asked, is DNA everything that matters? Well, according to what we know today, probably not. So there is also a broad class of changes um, called epigenetic changes, but so these are changes that almost literally mean on top of genetic changes, on top of DNA changes. So those are typically changes that don't modify the basic chemical structure of DNA, and that may also play a role in inheritance. But the jury on that is really out. There's a lot of um, tentative studies, and studies that turn out to be rather controversial in this field. So I think before I'd want to say anything definitive about this area, would probably have to wait another 10 or 15 years. So ask me
Mm,
then.
I see. it's it's interesting that, I mean, first of all, even Darwin's theory of evolution is kind of new in the broad span of history, and then I'm interested because of, to get deeper into your work then now, what is left sort of unexplained. Okay, there's random mutations, and that, there's we can almost measure, okay, how many mutations are there each generation. But how does, because we can always look around, and it's something that, there have been debates on, religious terms, why is there the complexity and the novelty that we have in the world? How has biological complexity emerged? So, tell me a bit more about how evolution actually, okay, like, we have the basics. How does it create the kinds of things that it does?
Yeah, so that's of course a very big question, and I'm not sure where to even start. Um, but
Okay.
well, actually, perhaps let's start with Darwin again. So Darwin was very puzzled by highly complex structures that he saw around him. So for example, take the eye. Somewhere in the Origin of Species, his most important work, he states that it's really a mystery to me how something as complex as the eye could have evolved by the kind of processes that I envision, namely natural selection. Um, because an eye is a highly integrated organ with many different types of tissues that all need to work together to accomplish a certain task, right? Um, but there's a lot of things he didn't know about the evolution of eyes. For example, now we know that eyes have evolved, maybe 20 or so different times independently from each other. That gives us an idea that, perhaps the evolution of eyes is not so difficult after all. Perhaps what we call innovation in evolution, and the eye, is a remarkable innovation for sure, is perhaps not all that difficult. Moreover, we know that multiple transitional stages, if you will, eyes of different complexity that give us an idea of how something as complex as the human eye could have evolved from much simpler parts. Okay? So that's one side. There's multiple origins of most innovations, and we know now a lot more transitional stages that were unknown to Darwin that kind of make a logical progression from something simple to the complex a whole lot more plausible, where all the intermediate steps could have been useful. And we don't just know this for eyes, but for a lot of other complex structures. So that's one class of problems associated with innovation. Another class of problems is the question of how a change in DNA translates into a change in an organism or in a trait of an organism. Because when we talk about DNA mutations, all we say is, well, there's a letter change or change in a structure of a chromosome, but it's not necessarily obvious how that change would translate into change on the level of an organism that we can see. And so this problem is also known as the genotype-phenotype problem. That is, the genotype is basically all the DNA of an organism, and the phenotype is what we can see, the organization, the structure of the organism beyond the DNA. And people also speak of genotype-phenotype maps. That is, those are conceptual or mathematical maps, if you will, that translate that show how changes in genotype translate into changes in phenotype. And that's really important to understand if you want to understand evolution, because it's about exactly the organization of that map. And again, coming back to Darwin, he knew none of this. The distinction between a genotype and a phenotype is actually was not even made until half a century after Darwin. And fundamentally, he couldn't make that distinction because he didn't know what a genotype was. He didn't know the material basis of inheritance and of variation.
Right.
Now,
Gone.
that being said, understanding this map is still a challenge today. And many of us, including myself, are most interested in looking at the kinds of complex biological structures where we can have some hope to understand this map and where we can use modern molecular tools to study this map. And that
It's gone.
basically brings us to one of our favorite study objects, and those are proteins. I'm sorry, Roy, you were going to say something?
Sure. It's interesting because we can get into both of these. I just want to like step back and think. Okay, so on the one hand, it's interesting that there are lots of innovations in biology that an eye is super complex. How does that come about? And it's weird to imagine, like, would all the pieces have to sort of come together at the same time, and then it's like that feels like an insurmountable problem. So that's one way in which something we need to explain, right? And then another you say, and it's something that Darwin again couldn't have really known, is this weird complex map between the DNA which changes, and then that impacts the phenotype, how the organism actually is, but it's not obvious how that happens. So that's okay, interesting. There's two kind of things here. And you were saying about you can then study phenotype, genotype in an interesting way. So why don't you go on with that?
Yeah, so um, let's look at proteins, for a moment. Proteins are basically the workhorse molecules of life. So basically, they do almost all the jobs that need to be doing inside all of our cells. Each of our cells probably has, um, you know, somewhere of order of perhaps a billion or so protein molecules, and these protein molecules come in tens of thousands of different kinds. Each one of them consists of a string of chemical building blocks called amino acids. So proteins are strings of amino acids, and these amino acids, how to make this string, is encoded in the genome. Now, um, so basically, in a gene, you have a DNA sequence, and that DNA sequence encodes a protein. But it's not so easy to predict what the properties of the protein, would be like if you just know the DNA sequence. And in fact, people have struggled with this problem. So it's a specific kind of genotype-phenotype map, the map from a protein genotype to a protein phenotype that people have struggled to understand for like half a century. Now, in the last 5 years or so, serious strides have been made with the help of AI, but the problem still remains challenging for at least for some proteins. And so, if you want to understand, for example, the evolution of proteins, let's take let's take again a bacterial protein because those are the ones that I study the most. Bacteria, for example, make proteins that allow them to be resistant against antibiotics. That is, the kinds of molecules that modern medicine uses in the hospital to combat or to fight bacterial diseases. So these proteins, we'll call them antibiotic resistance proteins, are encoded in genes in a DNA sequence. And from that DNA sequence, a amino acid string is produced. This string actually does not exist as a linear string inside a cell, but it folds up in rather complicated ways. And it is this folding, this folded state that allows bacteria to be resistant against specific antibiotics. Now, if we want to understand how this innovation, this bacterial innovation evolved, and um, it may look like a small thing to be resistant against antibiotics, but for a bacterium, it's a life or death matter, right? If they have the right kind of protein that allows them to cleave and destroy an antibiotic, that allows them to survive, and otherwise they won't survive. So if we want to understand how these kinds of proteins evolved, we need to understand how series of DNA changes led to these kinds of proteins. And that's an area that a lot of people are very interested in. The general field is called molecular evolution because we study evolution on the molecular level. And because of all the great tools we have today in molecular biology and genetic engineering, this allows us to study these changes. These tools allow us to study these changes in great detail.
It's to take so because we talked about there an innovation like this really important protein for fighting off different things, or an innovation like an eye. These without them, like for an organism like us, we kind of need it, and similarly for these bacteria, and you could think, well, there's so many different possibilities, say proteins you could make, and it's hard to know even what the map is between the DNA and what protein you're going to make. So would you say that there is something that one needs to understand about evolution? Like, how does it go and find that useful protein if there's so many different ones you could make?
Yeah, so there is different ways of thinking about this, and one way that that people, a lot of researchers approach this problem is to study what are called protein spaces or genotype spaces. Okay, so um, Let me explain that concept. Let's say we have a protein that is encoded by a string of DNA that's about 100 letters long. Now, you can think of this string as a text. Okay? This text is written in a four-letter alphabet. The letters are A, G, C, and T. And you can think of that a single such text as being part of a much larger collection. We might call it a library of such texts. Okay? And this is a really enormous library because if you have four possible letters at each of the 100 positions of that text, the total possible number of text is 4 times 4 times 4 and so forth until 100. So that's 4 to the 100th power, which is a very, very large number. And that basically would be constituting what we call a genotype space. Now, each of these texts could be conceived as encoding a different kind of protein. So basically, this library harbors the information to make all proteins that can be encoded in these 100 base pairs or nucleotides or letters. And imagine that we were able to understand the complete map from each of these texts to to the protein it encodes. Then we would have a complete understanding of this genotype-phenotype map. And it's also in this space where all innovation takes place, at least as far as proteins are concerned. That is, every thing you can do with a protein encoded by 100-letter text, is represented in that space. So just as a conceptual framework or as a metaphor, this is a very, very powerful idea because then we can ask, okay, so how would evolution actually explore this space? It might start out with one text string, and that text string then becomes a DNA string that would then undergo change through individual DNA mutations. And a lot of these might not lead to anything useful, but some of them might actually improve the protein that you started out with. And natural selection is very powerful at preserving such improvements. And from such an improved protein, you might actually then step through further mutations to yet another protein, and so forth, until perhaps you have found somewhere in this space a protein that could convey antibiotic resistance to a bacterium or any other protein that might be useful. So my lab and a lot of other labs around the world study these kinds of spaces, and they're very complex. They're much larger also in terms of their number of texts that they contain, DNA texts, that we could ever hope to map exhaustively. So we have to think about ways of getting some information on the organization of these spaces by sampling them in clever ways. But fundamentally, the idea here is that you can get some understanding about how evolution works by studying the structure and the organization of these spaces.
Right. So it's interesting that we see, an interesting innovation, an important innovation in a protein that a bacteria uses. But okay, how did they find this, so to speak? You could imagine you could actually, as you say, then actually try and be systematic about mapping out, well, what is the possible space that evolution is exploring, right? So, you can literally think about all the possible changes that could be made. And as you say, it's kind of astronomically large. But so you tell me about how you've tried to explore like that space and what that kind of tells us if we actually explore that sort of hypothetical space.
Mm-hmm. So, there's different ways of exploring these kinds of spaces. And one is computational, where you basically start with some kind of text string. Then you, in your computer, introduce a number of mutations to it. That is, some number, 30, 40 different individual single DNA letter changes to it. And then you use computer programs to predict the outcome of that change on what a protein might be doing. Or other, there's other molecules in the cell that are also encoded by DNA. It's not just proteins. For example, there is a class of molecules called RNA where we can predict the molecular properties much better in the computer. And so this is what in practice we work with, but I'll stick with the protein example because that's what we started out in. We started out with. So then you have 30 or 40 different mutant proteins. Let's say we can predict what they're doing, and that one of them is an improvement over where you started from. And then you take that one and you introduce another 40 mutations, and you ask what fraction of those improves the new protein, and you sort of um, keep doing that until you find a protein that is maybe much superior to what you started out with. So that's a computational approach to this problem, but there is another one too, and that's experimental. It mimics, in a sense, what we do in the computer, but it's of course the real thing because it works with real molecules, real proteins. And there, we might do this in the test tube or inside an organism. We again start out with a DNA sequence that encodes a protein, and then as we say, we mutagenize it. That is, we expose it to enzymes that change its DNA one letter at a time, introducing in this way a number of mutations. And when we do, some of the proteins that result will no longer have any function or be of any use to a cell, but others may have preserved their function, and yet others may have improved. And then again in the laboratory, we select for a specific property of a protein that we might be interested in. For example, conveying antibiotic resistance. We preserve those proteins, those mutant proteins that have improve their property or show some signs of having their property, and we then mutagenize them again and then expose them to selection again. So we then repeat this cycle of mutation and selection in the laboratory until we've found a protein that has all the properties we want. This process is also called directed evolution or experimental evolution. When it's done in a test tube, it's called in vitro evolution, but you can also do it inside an organism. And it's not just important for understanding fundamental principles of evolution and how nature might innovate, but it's also of huge practical importance. For example, biotechnology uses this very approach to engineer enzymes that are important in daily products. For example, if you do your laundry, then the detergent that you're using has a very high pH, that is very alkaline, because I don't understand the chemistry of that very well, but basically has to do with getting your clothes clean. And it works with enzymes that can operate at this high pH. And these enzymes are engineered, some of them through this kind of process.
It's a so it's interesting that you could actually create the kind of enzyme that you want with specific properties. Like imagine there's 5 things it needs to do, then you sort of keep selecting for one thing, and then all the things that survive that, and there might be kind of different ways of solving a particular problem, like this, it needs a certain shape or it's a certain property, and then out of all those you do it again and again until you have all of them. And so not only can you create useful things from it, it shows you the space of possible ways you could create this enzyme, this protein. And what does that tell us about evolution when you do these experiments and you try and explore this space really thoroughly? And because it's interesting, I guess, if we learn about this space, we can kind of then see what evolution is exploring, and that can kind of tell us, how it's, for example, even to go back to the thing we talked about earlier, like an eye, you might think it's really complex, but then it occurred 20 times in evolutionary time, let's say, in different places, right? So from different starting points. So it's almost like, what was the map that, evolution was exploring, so that it could get to eyes many places many times.
It's a very good question, and I'm glad you bring up the example of eyes again because it illustrates the merit of working with something simpler than a protein. In eyes, that space is just simply too complex. And for an eye probably has, millions of cells, and each of these cells you have, tens of thousands of different kinds of proteins. To understand that map is beyond our current ability of how, the genotype that encodes an eye maps onto that phenotype. But for proteins, we can do that. And what we and others have shown is something that is actually very reminiscent of what we see in these more complex organizations like the eye. And namely that most biological problems seem to have not just one unique solution, but many, many different solutions. So let me explain that. Um, by mapping these genotype spaces that I've talked about, either computationally or experimentally, what you learn is that if you take any particular protein that you might be interested in, let's say again an antibiotic resistance protein, and ask, where in this space, so where in this library of DNA sequences can we find sequences that encode such an antibiotic resistance protein? You might think, well, antibiotic resistance, that's a complicated chemical property. That is, a protein needs to be designed or engineered in just the right way to to cleave an antibiotic so it can no longer cause harm. So you'd think, well, maybe there's only a single such DNA text that encodes this property since it's such a rarefied and highly sophisticated property. But it turns out that's not at all the case. What actually is going on is that there is not only many different such DNA texts all over the library. There is in fact too many of them for us to count exhaustively because the library is so large. So not only is the library large, also the number of solutions to this problem, let's say of antibiotic resistance, is so large that we have trouble counting them. And these solutions are scattered all over the library, all over this genotype space. And that is precisely what allows organisms to discover the same kind of innovation multiple times, because simply as they explore through random DNA mutation and selection this space, they simply stumble upon one of these different solutions to the same problem, and different organisms stumble upon them in different ways and in different regions of this space. So coming back to the eye, if something like that is going on in the evolution of eyes as well, it becomes kind of unsurprising that evolution has discovered eyes multiple times independently of each other.
Mm, because even though the whole space of all possible, like say in the simple example of proteins, which we study, but what maybe in perhaps whatever different intricate things evolution has come up with, the space is huge, but also that means the number of possible useful things with the specific properties you want is also enormous. And then the thing that am I right in understanding one of the things you people studying this in the lab are kind of understanding is that the distribution of those useful innovations throughout that entire enormous space is such that they're distributed in such a way that you can get to them, that evolution can kind of find them from many different possible starting points. Or it's because
Yeah,
it's
so
if they're
that's
all in one. Yeah.
mhm. Yeah, so
It's
that's a very interesting point you're bringing up, and that's indeed correct. So let's imagine, that there were many solutions to any one problem, but they were all sort of bunched up in a small region of that space, right? And let's imagine that an evolving bacterium starts out in a completely different region because that space is so huge. In fact, much larger than any organism on this planet could have evolved since life's origin. Um, then it might never actually be able to find that solution. But what's actually turning out is that these many solutions to the same problem, they don't occur in the same region of that space. Instead, they're scattered all over the space. So wherever evolution starts out from, there is one solution to that problem nearby. And that's a really fascinating property of these spaces, that not only are there many solutions to most problems, these solutions are spread through the space, so it's relatively easy to find them.
Mm. It's it's what do you think about the existence of these spaces? I mean, I kind of want to, and maybe this is kind of unanswerable or almost philosophical, but what does it mean that? How can we explain that this space exists in the first place? Given that the distribution of this space and the fact that these innovations exist, across it, so they're not all bunched up.
Mhm.
Like why has that happened? Because
Mhm.
it's interesting because that feels like that's the fact that this space is distributed how it is. Without it, maybe life wouldn't have emerged, or emerged the way that it has. Right? It's only through the fact that there is this possibility space that is how it is, that evolution could really have kind of worked in the way that we've seen it, and could it have been different? And I'm kind of thinking about these kind of questions.
Yeah, so I think that it's a fascinating question. Unfortunately, we do not have firm answers to that question right now. Um, you know, so I could spend. I've
Mm.
been thinking about this question a lot. I could probably spend another half hour just speculating on what the reasons might be. Um, in the case of proteins, I think it's you know, a fundamental, if you will, property of protein biophysics, or even a sort of mathematical property of these spaces that actually, you know, has little to do with biology itself, in the sense that, even if if life had never evolved on this planet, the genotype-phenotype map of DNA to proteins, that are amino acid strings would still have a certain shape that is dictated by the biophysics of DNA and protein, right? But there is a much broader question beyond that. Is that the question why, um, is it that we have a particular type of DNA, or information carrying molecule, DNA, with its specific four letters? And why do we actually have amino acid proteins that are strings of 20 specific amino acids? People have explored in the laboratory alternative alphabets, that is, alternative
Mm.
molecular structures that could also serve to um, inherit or to transmit information from generation to generation, and they could also serve to encode useful molecules, uh, molecules like the proteins we have today, but also perhaps proteins with different kinds of amino acids, or molecules with a different chemical structure altogether. And I think it's a fascinating range of questions of whether the kind of molecular organization that life has today is what made evolution possible, or whether there is a bazillion other such organizations with different kinds of carriers of information and different kinds of molecules that function like proteins that could have worked just as well. And we basically, I mean, there's been a lot of speculation on that, but fundamentally we know very little about that. But I think it's one of the most important and most fundamental questions that one can ask about evolution and life's organization at large.
Yeah, it's fascinating that you bring that up. That DNA, because I would think, imagine there's some alien organism that didn't find DNA at first, but with similar, like, yeah, it's maybe even beyond biology, but there's similar mathematical underlying properties that would mean that maybe there's similar kind of genotype maps, so to speak, in whatever different alphabet you could create, such that there's that other possibility space would find innovations too, and you'd get some other alternative kind of evolution. And as you say, we can't necessarily answer these kinds of things, but I mean, do you
Mhm.
think that who would work on these kind of problems? Would it be mathematicians? What would you think we can find out about these questions?
Yeah, I think what it needs is certainly probably a solid understanding of mathematics, but you'd also need to have a solid understanding um, of the basic organization of life. So yeah, I think you need both kinds of skills. But then there is also, chemistry is really important here. So, for example, under the conditions in which life first evolved on this planet, probably under much hotter temperatures. The atmosphere also looked very different than it looked today, for example, because there was no oxygen in the atmosphere. What kind of alternative chemistries would be possible to create a DNA-like molecule that would function as well as DNA does today? So that to answer that question requires a lot of chemical knowledge, and I know there's a lot of people interested to answer that question. But I'm interested, but I'm not really an expert in this area, so I'm not going to speculate on that. Well, fundamentally, what you need is you need information on what looks like today. Second, you need information on possible alternative chemistries, and to understand the structure of the resulting genotype-phenotype maps, you need sophisticated mathematics and probably also sophisticated computation.
Well, that's really amazing and helpful to think about the sort of deeper implications of like trying to understand evolution at a very deep level. And just I wonder what the future of your work is. What kind of things are you thinking about? These things, or what have you explored? And would you like to explore?
Yeah, so um, I think we're still trying to get a better understanding of these maps, and we have done a lot of theory in, until about 5 to 10 years ago. Um, and my um, my conclusion from that theorizing, not only by the kind of theory that my lab did, but other labs too, is that we probably have plenty of theory right now, but what we really need a whole lot more is experimental work. We need experimental information on the organization of these maps, because theory and computational analyses like the ones I described are always a bit limited. For example, they rely on predictions of phenotypes from genotypes, and these predictions may not always be correct. In fact, they may often be wrong. And so, what do you do then if your predictions are not very good? Well, an experiment, experiments can provide the answer. And right now, we are using cutting edge technologies and developing these further in the lab to actually perform such experiments on a fairly high throughput scale, where we basically would create hundreds to thousands or millions of different genotypes and basically determine how well the proteins they encode function inside a cell, inside an organism. So that's easily explained, or it's just summarized, but it is actually a lot of technical work, a lot of difficult work you need to do. So that will take a good long time to get this type of analysis to a scale where I feel that we get a really good experimental overview over the organization of these spaces. And beyond what I've just told you, for example, that different solutions are spread solutions to any problem are spread through such a genotype space, and a few other things that we know. Beyond that, we don't actually know very much, and that's surprising. Given how large these spaces are, there's probably lots of new principles that are still await discovery that could probably fuel the career, of dozens, if not hundreds, of scientists.
Great. Thank you very much for speaking with me, Andreas.
Thank you very much for having me. Thank you for your interest in our work, and I'll be happy to chat again once we have made some progress
Yeah,
in the
great.
areas we talked about.